Okay, very good morning to my dear students. So now we will start our lecture of Science 2. First of all, I would like to share my screen to you. Yeah, we will start just now. Yeah. So, my dear students, on behalf of Bren Mumbai Municipal Corporation Online Education English Medium, myself Jayashree from Chinchauli MPS Malad West. Today I am hereby to take your lessons on Science 2 of class 9 as we have started a chapter number 16, Heredity and Variation. Today is our session 4 for this chapter. So first of all, we will take a quick recall of our previous portion. Now, what we have seen in our previous lectures, we have seen the topics inheritance, yeah, genetics, heredity, inherited traits and expression of traits. We have seen about the chromosomes, their structure of chromosomes, on the position of the centromere, how many types of chromosomes are there? There are 4 types of chromosomes that are metacentric, submetacentric, acrocentric, and telocentric. Yeah, and based on their functions, chromosomes can be somatic and the sex chromosomes. Okay. Then in the second session, we have seen about the DNA. What is the full form of DNA? Deoxyribonucleic acid. Yeah. So who have discovered this DNA molecule? Frederick Meiker. In which year? In 1869. And we have studied that DNA is present in all the organisms. For example, viruses, bacteria, all plants, animals, and human beings. So what is the function of DNA? The function of DNA is to control the functioning, growth, and division. That is reproduction of the cell. Okay. And it also bears what? Hereditary characters and is responsible for genetic transmission. And for this reason, DNA are called as what? Master molecule. What they are called? They are called as a master molecule. Then we have seen about the structure of DNA. What is the structure of DNA? Who has proposed the model for DNA structure? Watson and Crick. So what they have proposed that the DNA molecule is a double helix. It is just like a string. Okay, and it is made up of nucleotides. Each nucleotide is made up of an phosphoric acid, deoxyribose sugar, and a nitrogenous bases. Okay. So in DNA, what are the nitrogen bases? There are of 2 types, purines and pyrimidines. Purines are of 2 types, adenine and guanine. Pyrimidines are of 2 types, cytosine and thymine. Okay, the helicals have hydrogen bonds with the opposite sides. This is all about the DNA. Now what are the genes? Genes are defined as the functional units of the heredity, and genes control. What is the function of it? Genes control the structure and the function of the body and also help to transfer the information from one cell to another. What is the function of genes? Genes control the structure and the function of the body. Okay. Then we have seen about the DNA fingerprinting. What we have seen? DNA fingerprinting. Yeah. In this, what is the DNA fingerprinting? The sequence of the genes in the DNA of a person is called as genome, and this technique is used to identify the lineage and to identify criminals because it is an unique to every person. Then we have seen about the Human Genome Project. So what is a Human Genome Project? Human Genome Project is a great work in the field of human genetics, which has been done by the few nations. Okay. Then we have seen about the RNA. What is the full form of RNA? Ribonucleic acid. And this is the another important nucleic acid which is formed of ribonucleotide molecules, and it is also made up of ribose sugar, phosphate molecules, and 4 types of nitrogenous bases. They are same to DNA, but only one nitrogenous bases are different. That is adenine, guanine, cytosine, and uracil. Instead of thymine in RNA, uracil is present. Okay, my dear students, up till here you understand. And there are different types of RNA also. 3 types of RNA: rRNA, mRNA, and tRNA. rRNA means ribosomal RNA, mRNA means messenger RNA, and tRNA means transfer RNA. Then we have seen about the Mendel's principles of heredity. Here is an introduction of and scientist, that is Gregor John Mendel. And who was Gregor John Mendel? He was an Austrian monk or an Austrian scientist who studied the inheritance of traits in pea plants, and he developed the laws of inheritance by doing what researches on the plants, especially pea plant. And Mendel's work was not recognized until the turn of the 20th century, means up till 20th century his science, various researches were not seen by anyone. But in 21st century and after 20th century, his work was appreciated and recognized. Okay, so he was the person who is responsible for the principles of the heredity. Okay, now see the next slide. This picture is of a pea plant. This pea plant is Pisum sativum. What is the scientific name of the pea plant? It's an Pisum sativum. So this scientist, Mendel, what he has done, the genetic material which is transferred in equal quantity from parents to progeny, he has searched this. And which is that genetic material? He has researched. Okay. So Mendel carried out research in this direction and put forth the principles of heredity which are responsible for such inheritance. In the previous lecture, we have seen what is inheritance. Inheritance is the tendency to transmit the variations from the one generations to the next. Means what? The characters which are transferred from our parents to us. This is inheritance. Okay. So he has studied all these cases in the plant, that is pea plant. So all of Mendel's experiments were based upon the visible characteristics of the pea plant, that is Pisum sativum. Okay. Now you can understand which is a pea plant. So what he has seen. See in this slide you can understand. Yesterday I have explained you that dominant and the recessive character. Dominant character means with the characters which are directly transferred from our parents to us. What are the recessive characters? The characters which are not observed by us. Okay. So in case of pea plants, see this. What about the seed shape? In dominant it is round and in recessive it is wrinkled. You can see in the slide in these pictures. Okay. What about the seed color? In the dominant, what type of color it is having? Yellow. And in recessive, it is green in color. What about the flower color? In dominant, the color of the flower is purple. And in recessive, the color of the flower is white. You can see in the slide. You are able to see? Yeah? Yes. Then, what is the shape of the pod? In dominant, the shape of the pod is inflated. And in recessive, it is constricted. See, what is the color of the pod? In dominant, the color of the pod is yellow. And in recessive, the color of the pod is green. What about the flower position? In dominant, the flower position is axial. And in recessive, it's an terminal. Okay. What about the height of the stem? In dominant, the height of the stem is tall. And in recessive, the height of the plant is dwarf. So these are the researches which were done by the Mandels and he has observed in the pea plant. Okay. Moving to the further, in case of human beings, just now we have seen this all these characters which are which we are observing in the pea plant. But now see this dominant and recessive characters in the human beings. First character is eye color. In dominant, we can see what? Brown or black. These are the common colors which we are seeing in our human beings or in our surroundings. Yeah, but recessive, it's a blue color. We rarely see. Yes or no? Hair, curly hair, it's a dominant. Straight hair, it's a recessive. What about the chicks? Dimple chick, it's a dominant. And normal, it's a recessive. What about hand, right tendency? Most of the people and most of the human beings, they are what? Right-handed. And we see rarely the left-handed. So they are recessive character. What about the rolling of the tongue? In dominant means what? Most of the people or most of the human beings, they can roll their tongues, but some are there who cannot. So non-rollers. And what about the skin pigmentation? In dominant in our India, we can see mostly the normal skin color. Okay. And what about the recessive character? It's an albino. Okay. These characters which we can see in the human beings all are this. Okay. Now, moving further to the monohybrid cross. Now, what is monohybrid cross? The cross with only one pair of constructing characters is called as monohybrid cross. In the term only we can understand, we can see that what do you mean by mono? A single or a one. So the cross with only one pair of contrasting characters is called as a monohybrid cross. Okay. So in this experiment, let us consider the characteristics of plant height with a pair of contrasting characters, that is tall plant and the dwarf plant. Yeah, a common characteristic is plant height. That is, and height can be tall or it can be dwarf. Yeah. So moving further, see this. In the parental generation, P1 consisted of what? Tall and the dwarf one. Tall and the dwarf plants. And these plants were crossed with each other. So what we have seen, after crossing these plants, the F1 generation is obtained. Yes, F is, what is here? F is for the filial. All the F1 offsprings were what? Tall. Were tall, and none of them was dwarf. Dwarf means what? Small heighted. This is all about the F1 generation when the plants were self-crossed. Okay. Then what about the F2 generation? Means what? Mendel called tallness as a dominant character, while dwarf is said to be a recessive character, as it was not showing its presence. See, in the F1 generation, not a single plant is showing the dwarfness. Yes. So Mendel concluded that the factors responsible for inheritance of characteristics are mostly present in pairs. So now these factors are called as what? Genes. Okay. So the experiments and its results are shown in the Punnett square. So capital 2T letters pair of genes of one parent. Okay. And small 2T pair of a genes of another parent. So what about the F2 generation? So when this in F2 generations, when they are self-cross between the F1 plants, the F2 generations show the following gametes. So what is the genotype here? See, three plants were tall and only one plant is dwarf. So what ratio we have seen? 3:1. Means three tall plants and one dwarf plant. Okay. This is all about the monohybrid cross. See, this is an Punnett square. Now, parent generation P1, here the phenotype is what? Tall and dwarf. Genotype means what? The presence of the genes. Capital 2T means what? Tall, and small 2T means dwarf. What are the gametes here? Single T and small t. Now, first filial generation, what it shows? The phenotype, the most of the plants were tall. Okay. So, in the parental generation P2, selfing in F1, the crosses were done with the self. So, here the phenotype was tall and tall. Genotype is one capital T, one small t. What are the gametes? What are the gametes we are getting? One capital T and one small t. So what we have seen in the second filial generation F2, here we have seen that only one perfect tall plant is there. Otherwise, two tall plants are there, but they are not that so. Okay. So, and only one dwarf plants are visible in this progeny. Means one homozygous dominant and two heterozygous dominant and one homozygous recessive. Understand up till here? Homozygous dominant means capital 2 T letters. Heterozygous means one capital letter and one small one. Okay. And homozygous recessive means both are same. Both letters are same. This is all about the monohybrid cross. Okay. So what happens in monohybrid cross? He crossed a tall plant with a dwarf plant and observed how the traits are transmitted, the progeny, and calculated the percentage of tallness and dwarfness in subsequent generations. So, when a pure breeding tall plant TT was crossed with a pure breeding dwarf plant small tt, all plants were tall in the first filial generation, that is F1. But when such an F1 tall plant was allowed to self-pollination, both the tall and dwarf plants were appeared in the second generation of F2. Means what? According to the data collected by the Mendel, out of 929 pea plants, 705, that is 705 plants were tall and 224 were short. Thus, the phenotypic ratio of plants is 3 tall, 1 dwarf, and genotypic ratio is 1 is to 2 is to 1. Same T means what? Just now I told homozygous dominant, one capital T, one small t. It shows heterozygous, and both small t gametes, it shows homozygous recessive. So thus it can be inferred that in the F2 generation, phenotypically there are two types of plants, whereas genotypically there are three types. So my dear students, up till here you understand all about the monohybrid cross. Yes. Now moving further to the next topic, that is Mandel's experiment on dihybrid cross. Now, what is there in this? In dihybrid cross, what happens? Mendel brought about a cross between a pea plant producing rounded and a yellow colored seeds. See here, two characters are there. Yellow and yellow and yellow colored and the rounded seeds, and the pea plants with wrinkled and green colored seeds. Okay, what are the two characters? The plant which is having rounded and yellow color seeds of the plant and green color and the wrinkled seeds. So in this cross, when the two pairs of contrasting characters were considered, that is color of the seeds and the shape of the seeds, hence it is called as an Dihybrid cross. Why it is called as a dihybrid cross? Because when the two pairs of contrasting characteristics are under consideration, then it is called as an dihybrid cross. So, round and yellow seed bearing plant was crossed with a green and wrinkled seed bearing plant of to have a dihybrid cross. So, round and yellow are the dominant characters, green and wrinkled are the recessive characters. Up till here you understood? Yeah. Now moving further. Now, Mendel's experiment of dihybrid cross. Now, what is the parental generation? It's a P1. Now, first parent, it is round seeds. So, it is indicated by capital 2Rs. Yellow seeds, it is indicated or it is given the name capital 25. Second parent, wrinkle seed, it is represented by a two small letters of R and two small letters of Y shows the green color seeds. Up till here you understood? Now, then, what are the gametes from RRYY? Means total capital letters parents means RY, and the gametes from small r, small y, X and small r, small y. Okay. So now, here the genotype for the rounded yellow seeds is this one. For the wrinkle green seeds, it is this. What are the gametes we are getting? For the round yellow seeds, this, and for the wrinkled, this. Now, the first filial generation F1, what it shows, we get. For this, the genotype of these plants was capital R, capital Y, small y. So their phenotype was producing what? Rounded yellow seeds. What? rounded yellow seeds. In the first generation, what we get? Rounded yellow seeds. Now, moving further to the parental generation, P2 now. Here, F1 means what? All the plants formed show round and yellow seeds. Yes. So, what is the genotype? All are having heterozygous, that is RRYY. So, the offsprings obtained through such a cross is called dihybrid. So the plants with RRYY produced four kinds of combinations of gametes. That is both capital R and Y, one capital, one small, one capital, and one both are small. Okay. From the rounded yellow sets, what we have seen here also the same one. So four gametes we have seen here. So when we form an Punnett square, what we observe? When we will form and Punnett square means the parental generation when the self cross were done. What is the phenotype here? Rounded yellow seed is crossed with the rounded yellow seeds. Then what happens? What is the genotype here? Capital R, capital Y, capital R, capital Y. What are the gametes here? Four gametes we have formed. So what is the second filial generation is shown here. Here we get, see how many this type of yellow, purely yellow rounded cysts we are seeing. See here, 1, 2, 3, 4, 5, 6, yeah. Then 7, 8, 9. So round and purely yellow seeds are how many in numbers? 9 are there. Round and green seeds now. Round and green seeds. 1, 2, 3. Total 3. Round and green seeds. Now 3 wrinkled and yellow seeds. Now you can see in this. 1, 2, 3. 3 wrinkled and yellow seeds. And what about the wrinkled and the green seeds? Only one, which is pure, means homozygous recessive. Here only we are getting one. So what type of phenotypic ratio we are seeing here? 9 is to 3 is to 3 is to 1. Now, moving forward, use your brain power. Now we can study about the phenotypic ratio and the answer. Yeah. Now see, just now we have counted round yellow seeds, total number of 9. Wrinkled yellow, what is the number? 3. Round green, what is the number here? 3. What about the wrinkled green? It is 1. It is 1. So this is all about the phenotypic ratio. What about the genotypic ratio now? Use your brain here. What is the genotypic ratio? Now see, here the numbers are given. See, purely capital means round and yellow. How many? Pure dominant, only one. Then pure recessive, you can see in this chart. See this, all are small in the last number. So it is a pure recessive. Okay. This one, see, round and yellow. How many? 2. Round and yellow? 1. In this way, you have to find out the genotypic ratio. You can understand from here. Means what? Round and yellow, 2 are there. Yes. Then round and green, 1 is there. Yes. Then wrinkled and yellow, how many are there? 2 are there. Then round and yellow, you can search here. So from this gamete formation only, you can identify their what? Genotype. So up till here you understood. So on this basis, what we can do? We can distinguish between the monohybrid and the dihybrid cross. So now what is all about the monohybrid inheritance and the dihybrid inheritance? Now we can see here, the first point is what it is. Inheritance. In monohybrid, the inheritance of a single allele pair. Here only single pair is done or crossed. In dihybrid, inheritance patterns of two pairs of characters during a dihybrid cross. In monohybrid, only single character, single pairs of characters, and in dihybrid, it's two pairs of characters. What is monohybrid inheritance? It is defined in the Mendel's first law. Okay. Monohybrid inheritance, it is defined in the Mendel's first law. But dihybrid inheritance, it is defined in the Mendel's second law. In the Mendel's second law. Then in monohybrid inheritance, the phenotypic ratio of the F2 generation is 3:1. 3:1. And in dihybrid, the phenotypic ratio of the F2 generation is 9:3:3:1. What about the genotopic ratio in the monohybrid F2 generation? It is 1:2:1. And in dihybrid inheritance, the genotypic ratio of the F2 generation is 1:2:1, 2:4, 2:1, and 2:1. Means total numbers you can add, you can get. Now, monohybrid inheritance, how it is used? It is used to study the pattern of inheritance during the first and the second generation offspring. Okay, where it is used, this monohybrid inheritance, it is used to study the pattern of the inheritance during the first and the second generation offspring. Now, what about the dihybrid inheritance? It is used to study the inheritance pattern of dominant and the recessive characters for two different traits. So, where this dihybrid inheritance we can use? This is used to study the inheritance pattern of dominant and the recessive characters for two different traits. So, my dear students, yesterday we have seen the monohybrid cross, and today we have seen in detail about the what? Dihybrid cross. So, we will take a quick recall of today's lecture, what we have studied with the help of these slides only, so that you can understand. And I want that if you want, you can take a screenshot from this, so that it can be easier for you to study or to revise the lesson. Okay. Now, today we have seen about the Gregor John Mandel, who was an Austrian scientist. He studied the inheritance of tracts in a pea plant, and he developed the laws of inheritance. Yes. Then in Mendel's work was not recognized until the turn of the 20th century. Then he has performed all his researches on this plant, that is pea plant. What is the scientific name of this plant? Pisum sativum. Yes, here the genetic material is transferred in equal quantity from parents to progeny. So Mendels carried out research in this direction and put forth the principles of heredity, which are responsible for such inheritance. And all of the Mendel's experiments were based upon the visible characteristics of the pea plant, that is Pisum sativum. Okay, then we have see in the slide, you can observe the dominant and the recessive character in a pea plant. You can see the dominant one and the recessive. Round, wrinkled, yellow and green, flower color, purple and white, pod shape inflated, constricted. What about the pod color, yellow and green? What about the flower position? In dominant, it's an axial, and in recessive, it's an terminal. What about the stem height? In a dominant, it is tall, and in recessive, it is dwarf. So it can be asked in the exams, all these characters in a tabular form to complete the table. Okay. Now we can see there the slide about the dominant and the recessive character in a human being. Yes, we can see. So see here the characteristics of the human beings. See eye color. In dominant we can see brown or black, and in recessive we can see only blue. Okay, what about the eye size? Large in dominance, small in recessive. What about the hair? What type of hair? Curly hair we can see in dominant, and straight hair we can see in recessive. What about the cheek? Dimple cheek we can see in dominant and normal in recessive. What about the hand? See, most of the people we see, they are right-handed, and most rarely we can see the left-handed persons. Yeah. Rolling of the tongue. Mostly the persons they can roll their tongues, but in recessive, most rarely the persons are having non-rolling tongues. Okay. What about the PTC testers? Tester and non-tester. Then skin pigmentation. The skin color in dominance is normal, but in recessive, it's an albino. Okay. This is all about the human being. Then we have seen about the monohybrid cross. What is monohybrid cross? The cross with only one pair of contrasting characters is called as what? Monohybrid cross. Here only one pair of contrasting character is considered, and this experiment let us In this experiment, he has considered only one character, that is plant height. Okay, with a pair of contrasting characteristics, that is tall plant and a dwarf plant. Okay, now here this is the what? This is the presentation of the crossings. Okay, so if after this self crossing, what happens in F2 generation? What we have got, that three tall plants and one dwarf plant. So what is the ratio here? 3:1. Then you can see in this Punnett square, you can see the genotype, phenotype, and the gametes. What are the phenotypes? The naming, tall and dwarf. What is the genotype? This these are the terms, T and small t, and the gametes which you are getting from here. This. So in the first filial generations, we get mostly the tall plants. Then in P2 generations, what is the phenotype? Tall, tall. In genotype, what is this? And these are the gametes. So what, with the help of the Punnett square, what we have seen in the second filial generation, that three are tall and one is dwarf, but only one is purely homozygous, what? Dominant, but two are heterozygous dominant and one is recessive. Yeah. Okay. Now, what is monohybrid? We have seen that he crossed a tall plant with a dwarf plant and observed how the traits are transmitted and the progeny, and calculate the percentage of tallness and dwarfness in subsequent generations. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, all plants were tall in the first filial generation. When such a F1 tall plant was allowed to self-pollination, both the tall and the dwarf plants were appeared in the second generation. Okay, so according to the data collected by the Mendel, out of 929 pea plants, 705 were tall and 220 pair were short. Thus, the phenotypic ratio of this plant is what? 3 is to 1 is to 1. Okay. So, what is the genotypic ratio? Is 1 is to 2 is to 1. Okay. Thus, it can be inferred that in the F2 generation, phenotypically, there are two types of plants, whereas genotypically, there are three types of plants. Then we have seen about the dihybrid cross. Here, in this cross, two pairs of contrasting characteristics were considered, that is color of seeds and the shape of the seeds. Hence, it is called as a dihybrid cross. Then, see here, you can see the phenotypic, genotype, the gametes which we have seen here. So, in the first filial F1 generation, the what phenotype we got? Rounded yellow seeds. Rounded yellow seeds we have seen in the F1 generation. Now, in the parental generation P2, when the rounded yellow seeds were crossed with cells, then what the gametes which we have got? We have got four gametes. So second familial generation with the help of the Punnett square, we can see here. See how many plants we have get. So in further slide, we can see the phenotypic ratio here. See the round yellow, it's of 9 number. Wrinkled yellow, it's 3. Round green, it's 3. And wrinkled green is 1. So what is the genotypic ratio of this? See, pure dominant is what? Round and yellow. And pure recessive means what? Round and not round, wrinkled yellow. Okay. So this is all about the dihybrid cross. So in this way you can do. So, and you have to take this screenshot of this distinguish between monohybrid and dihybrid cross. In exams, it can be asked. Differentiate between distinct monohybrid and dihybrid cross. So in this way, you can differentiate this both crosses. Okay. One time I can repeat this. What is monohybrid inheritance? Inheritance of a single allele pair. Dihybrid inheritance. It's an inheritance pattern of two pairs of characters during a dihybrid cross. And what is monohybrid inheritance? Defined in a Mendel's first law. And dihybrid inheritance is defined in a Mendel's second law. Then in monohybrid cross, phenotypic ratio of the F2 generation is 3 is to 1. In dihybrid inheritance, phenotypic ratio of the generation is 9 is to 3 is to 3 is to 1. Genotypic ratio of the F2 generation in monohybrid is 1 is to 2 is to 1. In dihybrid, its
Genotypic ratio of F2 generation is 1:2, is to 1, is to 2, is to 4, is to 2, is to 1, is to 2, is to 1, means total 9 types of. So, in which reason or how it can be used? Means who? Monohybrid inheritance. It is used to study the pattern of inheritance during the first and the second generation of spring. Yes, and in the hybrid inheritance, it is used to study the inheritance pattern of a dominant and the recessive characters for two different traits. So, my dear students, by here I am concluding my today's session, and in the next session we can see about the some human characters, okay, and the genetic disorders also. So, by this I am concluding my lecture, and I am very thankful. So by this, we have finished our lecture now. Yes. So I would like that my students should read the lesson thoroughly and write the answers and of the questions which are given in the exercise. Read it and learn it. So by hereby, we have completed our today's session. Now I am ending this lecture. So thank you students for listening me, and we have completed our today's 4 lectures. Okay.